So with that, our next presenter is going to be Dr. Francisco Penñagaricano, who is an assistant professor at the Department of Animal and Dairy Sciences at UW-Madison. He's broadly interested in quantitative genomics and computational biology. His research program focuses on developing and applying methods to dissect the genetic basis of economically relevant traits in dairy cattle. His research involves gene mapping, genomic prediction, multi-genomic data integration, and network modeling. And we also want to point out he has a very active website as well. I'll drop in the chat box. But with that, we're very happy to have Francisco with us. He's doing some really cool things, and we'll turn it over to you.
Thank you, Ryan, for the introduction. Can you guys see my slides?
Yes, we can see that.
Excellent. Thank you very much, and thank you all for joining us today. And I will be talking about cow fertility in the post-genomic era, and I will go over some progress, challenges, and some perspectives. But let me start with what's the goal of a selection program, right? Well, our goal is to improve either traits that increase revenues or traits that reduce expenses. And as you can see here in this table, we have many traits under selection in dairy cattle, including production, fertility, longevity, health, calvinability, conformation, and even sustainability with the new National Genetic Evaluation for Feed Efficiency. And we do believe that fertility is a very important trait, but for us, geneticists, fertility is one more of the traits that we have under selection. Now, let me start with a very successful story. That is the change in milk production in the last 60 years. Here I'm plotting milk production per cow per lactation in the y-axis over the last 60 years. And as you can see here clearly, there has been a tremendous increase in milk production per cow per lactation. Actually, dairy cow nowadays, she produces more than twice as much milk as a cow 60 years ago. And more than half of that improvement is due to genetic selection. In fact, genetic selection is a very powerful tool for achieving lasting gains in performance, and contrary to improvement in nutrition, management, cow comfort, the changes achieved through genetic selection are incremental, cumulative, and permanent, what makes genetic selection a very cost-effective strategy. Let me tell you another successful story, that is the change in dairy cow fertility in the last 60 years. I'm still plotting the change in milk production in the last 60 years in blue, and I'm plotting now the change in reproductive performance, measured as pregnancy rate, in green. And as you can see clearly in this graph, concomitant with a very intensive selection for milk yield, pregnancy rate declined steadily until the beginnings of the 2000s. The good news is that we were able to stop the decline, and actually we have observed a tremendous increase in reproductive performance in the last 10 and 15 years, as you can see here. In fact, a dairy cow today has the same fertility as a cow in the early 1980s. I'm plotting now in green the change in reproductive performance, but I'm plotting in brown now the change in the genetic merit of the animals for fertility, including the sires here and the cows. And as you can see clearly, there has been a quite important deterioration in the genetic merit of the animals until the end of the 2000, roughly 2008, 2010. And actually was because the use of genomic selection that we could stop the decline in the deterioration of the genetic merit of the animals. And in fact, the genomic merit of the animals are slowly but steadily increasing now. I think it's fair to say that Part of the decline in reproductive performance was driven by the deterioration in the genetic merit of the animals, but probably most of the part of the increasing reproductive performance that we have observed in the last 15 years is due to non-genetic factors. Before I keep going, let's review what are the traits that we have under selection nowadays. And we have actually 4 different traits: daughter pregnancy rate, heifer conception rate, cow conception rate, and early first calving. Daughter pregnancy rate reflects cows' genetic ability to conceive sooner after calving. And that was a trait that was developed in 2003. We have 2 different conception rates, one for heifers and one for cows. And these traits reflect heifers or cows' ability to conceive. And these 2 traits were introduced in the national evaluation in 2009. And 3 years ago, we developed a new national evaluation for a new fertility trait, early first calving, that basically reflects heifers' ability to calve early. It's measured as age at first calving. I'm showing here the genetic trends for fertility traits in the last 20 years. And I'm plotting the change in the genetic merit of sires and cows. And I have daughter pregnancy rate here. In the middle, I have heifer conception rate. And I have here cow conception rate. And as you can see here clearly in this plot, we observe a deterioration of the genomic merit of the animals until 2008, 2010. That is exactly when we start to use genomic selection. And nowadays, the genomic merit of the animals for fertility traits are slowly but steadily increasing, as you can see in this plot. You know, genomic selection is the latest revolution. It has transformed dairy cattle breeding, and it has caused the major or the most remarkable change in dairy cattle breeding since the introduction of the artificial insemination. Genomic selection is based in a very simple but elegant concept, that is the use of genetic markers across the genome to predict breeding values. And because we are using the genome of the animal to predict the breeding value of that animal, this method allows us to detect animals and at early age. And that is very important, especially when we are talking about dairy cattle breeding, where all the relevant traits are sex limited. All the relevant traits are expressed in the cows and cannot be measured until these cows begin lactating. So if we have a tool that allows us to select animals and at early age, that will have a major impact, especially in dairy cattle breeding. This is to show you that genomic selection is actually a tool used by farmers here in the United States. I'm not talking about nothing strange, nothing magical. This is the number of genotype from Holstein animals that we have in the national evaluation. And today we have close to 5.5 million genotypes. And as you can see here, I'm plotting since 2011, this has been an exponential growth in the number of genotypes. The other thing that is quite interesting, that the vast majority of the genotype, as you can see here, they come from females. They do not come from males. Most of the genotypes that we have in the national evaluation are from heifers from commercial farms, as you can see here. A very small proportion of the genotypes, they come from males. So this is a tool that is used by commercial farmers. What's the beauty of genomic selection? Well, the beauty is that genomic increases reliability, and reliability is how accurate genetic merits are estimated. And think about this. We have to estimate as precisely as possible the genetic merit of a newborn heifer or a newborn bull. It's quite a simple exercise, but it's not easy. If we do not have any other information, our best prediction will be based on what we call the parent average. That is the average of the sire and the dam. And if we make selection decisions based on parent average, the reliability of that prediction ranges between 0 and 0.35. 0 basically when we don't know the pedigree, so there is no way to have a precise parent average, or we believe that we know the pedigree, but the pedigree is wrong. And in cases that we have a very complete pedigree, the reliability is up to 35%. Now, if we need to estimate as precisely as possible the genetic merit of a newborn heifer, and what we do is actually take a sample of that animal and genotype the animal using genomic testing, then the selection decisions will be based on the breeding values, the genomic breeding values, the breeding values estimated based on the genome of the animal, and that's reliability, depending on the traits, range from 0.65 to 0.80, and it's actually this difference, reliability is between 0 and 35%, and reliability from 65 to 80%. That is reflect the how powerful is genomic selection, genomic testing in dairy cattle. And of course, if we can get higher reliabilities, we will achieve higher annual genetic gains. I'm showing here a comparison between genomic reliabilities versus traditional, or those reliabilities based on parent average. And this is data from the last evaluation a few weeks ago. This is data using 3,340 Holstein bulls. I have many traits here, but please focus in this very last column that I have here in yellow, that I'm comparing the genomic reliability versus the parent average or the traditional reliability. And as you can see here, the difference is always positive. For production traits, we have difference between 46, 47, but then for longevity and fertility, we have larger differences around 49, 50, and 53. So genomic selection has a very positive impact in all the traits, but the gains are greater for fertility and longevity traits. In fact, genomic selection, using genomic selection, we have achieved twofold changes in production, but even 3 or 4 changes in fitness traits like fertility and longevity traits. Again, genomic selection has an impact on all the traits, but mostly, or the benefits are greater for fertility and longevity traits. So a question that maybe you are thinking of is, well, does genomic works? In other words, can genomic testing predict future performance, right? Because we are able to genotype a heifer, and then is it possible to predict the performance of that animal in the future? And given that today we are talking about fertility, I will show you this result about predicting reproductive performance using genomic testing. This is a study that we published a few years ago in the Journal of Dairy Science. We used data from commercial farms, 4 farms in the state of California. And in this plot, I'm using data from more than 3,000 primiparous cows. Each of these columns has roughly 850 primiparous cows. So let me tell you what we did. We genotype all the heifers at weaning, and we calculate the genomic DPR. And we group the heifers in 4 groups, from the top 25%, the most fertile animals, to the bottom 25%, or the least fertile animals, based on the genome. We didn't do anything, and we wait until the first lactation, where we measure the actual reproductive performance. And I'm plotting here the relationship between pregnancy rate of first insemination versus the quantiles for genomic DPR. And as you can see here clearly, the group that we say probably 2 years ago at winning, this is the most fertile group. This is the group that achieved the highest pregnancy rate at first insemination, while the group that we say these are the least fertile animals based on the genome is the animal that achieved the lowest pregnancy rate at first insemination. Very cool results. We did the same now, but trying to find a link between genomic DPR and number of services per conception. The same concept here. I have 4 groups of animals based on genomic DPR. And as you can see clearly here, the most fertile group, the top 25%, need the least number of services per conception, while the least fertile group, or the group with the worst genomic DPRs, was the group of animals that need the greatest number of services per conception. And the same concept here between genomic DPR and the interval between calving and first insemination. The most fertile group based on the genome, and again, this prediction was done at winning. That group showed the shortest interval between calving and first insemination, while the least fertile group, or the worst group in terms of genomic DPR, that was the group that showed the longest interval between calving and first insemination. Extremely cool result showing the relationship between genomic testing and reproductive performance. Again, does genomics work? Can genomic testing predict future performance? Well, this data is showing us that genomic testing can be effectively used to predict performance. I haven't showed this result, but as you expect, with genomic testing, we can make a more accurate prediction that using just desired PTA values. And because we can effectively use genomic testing to predict performance, it can be used to make proper selection decisions. And because we can do it at an early age, few weeks after birth, if you want a weaning, and we have a tool that can be used to predict performance, now we can use that tool to make proper selection decisions. So this is a quite interesting question, how fast we can move in terms of fertility rate. Well, in general, how fast we can move for any trait. As you can see here, this is the change in pregnancy rate in green, in reproductive performance. And I have here the change in the genetic merit of the animals for the 3 traits, DPR, heifer conception rate, and cow conception rate. And as you can see here, genetic merits are now slowly but steadily improving, which are great news. And let me tell you again, genomic selection was a key tool to stop the decline in the genetic merit of the animals and actually to start to move those genetic merits up. Now, how fast can we move? Well, that depends on the selection tool that we use. And the selection tool that we use is a selection index. And here I'm showing different selection indices. We have net merit, very famous. Fluid merit, quite popular, for example, in Florida, in a fluid market, or cheese merit, quite popular here in Wisconsin. There is no discussion that the selection index is the best selection tool that we have. Now, if you check these 3 indices here, regardless the index, fertility receive a weight of roughly 6.5% only. And that somehow limits how fast we can move. I told you in my first slide, fertility is a very important trait, but we have many traits under selection. And for us, fertility is one more of the trait under selection. Now, if you take a bigger picture, if you see the entire picture, the entire photo, you will see that fitness trait, and I'm talking now together about fertility, longevity, and also health. fitness received quite a large weight in our selection indices, roughly 34%. And this weight is probably supporting the dramatic improvement in reproductive performance that we have observed in the last 15 years. This one is quite important question, right? We have TPR, heifer conception rate, and cow conception rate. Are these traits good fertility traits? And before we start to criticize these traits, let me tell you that these traits can be easily measured on many cows. What for us, the genetics is quite important. The plots that I have been showing you guys are based on roughly 700,000, 800,000 cows per year in each of the dots. So again, these are traits that we can easily measure on many cows, and that's very important. Now, at the same time, these traits are lowly heritable. Conception rates, they have a heritability around 1–2%, while daughter pregnancy rate has a heritability up to 4%. Probably these traits are distant from cows' reproductive physiology. And what is even more important, these traits are impacted by management, including voluntary waiting period and synchronization protocols. And today I want to touch about these synchronization protocols. Synchronization protocols are great management tools. I'm not here to criticize the use of synchronization protocols, but you will agree with me that synchronization protocols somehow mask for cows' fertility ability, right? Because genetically superior and inferior cows may show similar phenotypes. And as you know, genetic programs rely on the collection of accurate phenotypic data. And if we are collecting data from cows that are treated, those records may bias genetic evaluations. So this is a very important question, a very hot topic nowadays, actually. And what's the impact of the use of data from synchronization protocols on genetics evaluations in dairy cattle? And the group of the University of Guelph in Canada actually tackled this question a few years ago. And what they did is they used data from the Canadian evaluation, and they have basically 3 different fertility traits: time from calving to first service, service to conception, and days open. And basically they split the whole data set in 2 different data sets. A data set with record from synchronization or TIAI protocols, and data set that is based on record from heat detection protocols. And what they did is they ran a genetic evaluation in each of these 2 subsets, and they estimate the sire's PTAs for these different traits, and then they basically evaluate the re-ranking. If the correlation between sire's PTAs in these 2, based on these 2 data sets, are quite large, that means there is almost no re-ranking. Probably there is almost no impact of synchronization protocols on the estimation of sire PTAs. Basically, doesn't matter if the cows of the daughters are evaluated under time AI or heat detection, the evaluation are almost the same. Now, if there is a large re-ranking, probably that means that there is an interaction between the reproductive management and the genome of the animal. And actually what they found is that there is a large re-ranking. They got low correlations between this sire PTAs, and this is across trait and across parities. And these results suggest that synchronization protocols may bias genetic evaluations, and the solution is not clear. Probably what we should do is to consider reproductive performance under synchronization protocols or time AI protocols versus heat detection as different traits. And in that way, we will have PTAs for desires under tiny eye or under heat detection protocols. This is, I think, a quite interesting question nowadays. Do we need new fertility traits? And you guys are the expert, but maybe we need traits that more closely describe cow's reproductive physiology. And I'm showing here a set of different events where we can evaluate fertility, from resuming cyclicity postpartum to expressing estrus, ovulating, conceiving, establishing pregnancy, and maintaining pregnancy. We would like to have traits that have a decent heritability. And in the best scenario, we would like to have traits that are not affected by management practices. The question is what to measure? Well, you know, we could measure physiological traits like follicle count, or anti-Mullerian hormone, or progesterone concentration, or pregnancy-associated glycoproteins. Or we could even measure some anatomical traits. And I'm thinking about reproductive tract, size of position, or anogenital distance. It's interesting, for these anatomical traits, we do not understand exactly the relationship between these traits and reproductive performance. But these anatomical traits, they tend to have larger heritabilities. So we could actually improve faster these traits in the population. Now, of course, we are talking about novel traits, but this is a great opportunity in the genomics area, because genomics has created opportunity to improve these traits that are critically important, but are too difficult or expensive to measure on the entire population. Imagine, for example, measure progesterone or whatever you can imagine. What we can do is we can measure these relevant phenotypes in a very small reference population of cows. We will also genotype the cows, and through prediction, through genomic prediction, we will be able to estimate genomic PTAs for the entire population, including young selection candidates. Please note that we have a current example of that, and actually, dairy cow feed efficiency, the national evaluation for feed efficiency, is based on phenotypes that we collect in research farms, including our farm here at the University of Wisconsin-Madison. So this is nothing crazy, nothing strange, nothing magical. We have already a very successful example where we are evaluating a quite important trait, a novel trait, using phenotype in a small reference population. My group has been working on this novel fertility trait. We are, for example, evaluating estrus expression, and we are phenotyping estrus expression using activity and rumination monitoring system. We believe that if estrus expression has a genetic component, then we can improve estrus expression through genetic selection, and we will have a better expression of estrus by the cows. We will have a higher estrus detection by the personnel in the farm, and at the end we will have a higher insemination rate and also pregnancy rate without the need of using reproductive hormones, and that probably will have a great impact on what we call sustainability of the dairy industry. We have some good news in this field. Estrus expression traits are heritable, what is quite important. Estrus duration around 8 and 10%, and estrus strength even larger, between 10 and 15%, and estrus expression rate are repeatable. So maybe a few number of records per individual would be sufficient. We are also working on fetal loss. Again, you are the expert. You know, fetal loss has a major economic impact, and it's also a quite important welfare issue. And our results show that fetal loss is a heritable trait. and the magnitude of the heritability suggests an important scope for selection. We do not have a national evaluation for fetal loss, so this could be a novel fertility trait. Fetal loss is largely independent of current fertility traits like daughter pregnancy rate. That means that current selection efforts have little impact on pregnancy maintenance. And we have shown that genomic prediction of fetal loss is feasible, what is quite important. And this provides potential for accurate genome guide decisions. What about functional genomics, or very basic research in genomic? Is it worth? And I'm talking about mapping genes or mapping causal variants or trying to find genes underlying trait. And these are our results. And here we found 5 regions in the Holstein genome that have a major impact on bull fertility. These are genomic regions that have a very strong impact on bull fertility. And the answer is yes. It's quite important to keep working on this. In fact, through functional genomics or research on functional genomics, we were able to identify and now track non-recessive defects that have an impact on reproductive performance, like DAMS, CMV, brachyspina, among others. And we were able to identify and track haplotypes that affect embryo loss. And these are haplotypes that have quite important frequency in the population, but are never observed as homozygous, right? Because they cause embryonic loss. And we have nowadays genetic tests that are routinely used to identify carriers. Actually, when you genotype your favorite heifer, you will get as result also information about recessive alleles and also haplotypes. And of course, if we have even more information about causal variants, we can include those in commercial SNP chips somehow to try to improve the accuracy of the genomic PTAs, or actually try to use models that include this biological information. And we strongly believe that if we have more biology or better understanding of biology, that could impact not only management, but also get more accurate predictions. This is my very last slide. This is the change in inbreeding. This is in Holstein in the last 60 years. And as you can see here, there has been a tremendous increase in inbreeding, mostly at the last 12, 13 years, that because due to the implementation of genomic selection. Should we be concerned? The answer is probably yes. We are not sure what will be the impact of the loss of genetic diversity on fitness trait like fertility trait. And I want to finish with some take-home message, and then of course I will be open for questions. Genomic selection contribute to stop the decline in dairy cow fertility, and actually genomic merit for fertility trait are now slowly but still improving. And I think it's important to remember that selection indices, the best selection tool that we have, consider multiple traits, not only fertility. Farmers do not select only for fertility, right? And if they do, they are probably making a mistake. Farmers select for multiple traits simultaneously. As I show you, genomic predictions can effectively predict future performance. I show you data from the field. What is very important about fertility and reproductive performance is that the genomic testing allows also to tracking of genetic recessive effects and haplotypes, and genomics could facilitate the selection for novel traits. We have a clear example, dairy cow feed deficiency, but we could think about several novel traits related with fertility. This is quite important. the potential interaction between genetics by repro management, for example, heat detection versus the use of synchronization protocols, deserves attention. And how to balance rapid genetic progress as we observe and as we want. Another way, genetic diversity is not a very easy question. And of course, functional research, or very basic research, benefits applied selection. With that, thanks for your attention. Thank you again for inviting me, and of course, I'm open for questions. Thank you.
Great, really good stuff. Thank you. And yes, if you have questions, please drop them in the chat. As you were going through there, a couple questions did come through. So I think, I'm trying to think the best way to articulate this. Maybe in our older thinking, we try to draw correlations between reproductive efficiency and increasing milk production. You couldn't have them both together, it was one or the other. As we've dug more into the genomics of this, is that really true? Can we have the best of both worlds? Or do those two traits work against each other?
Absolutely. Can you see my slide, Ryan?
Yep.
In the last 15 years, we observed the best of both worlds. We are still improving in terms of milk production, as you can see in blue, and we are improving in terms of reproductive performance. So the answer is yes, for sure.
Right,
and we can achieve that using a selection index that simultaneously will select for more milk and better fertility.
No, that's good. That helps. And then I don't know how far you've gotten into this, as you're talking about the different potential reproduction traits to start including investigating in the genomics world. Do you have any favorite contenders of which ones look the most promising? Is that still an ongoing area of research?
That's a very good question. So we are working at the same time in the two extremes. We are working in estrus expression, right? And we are working in fetal loss that is related with the pregnancy maintenance, right? Pregnancy maintenance has a huge economic impact. Abortion or fetal loss they have a huge economic impact and are a welfare issue. And what is interesting is it's a trait that is not well captured by current fertility traits. In other words, if we try to select for DPR, we are not changing much pregnancy maintenance. So that will be a very important trait to include in the national evaluation, because with the trait that we have nowadays, we are not improving that important area that is pregnancy maintenance. The second one is estrus expression. I think estrus expression is quite important. If we believe that in the near future, we will need to maybe minimize the use of reproductive hormones because the pressure of the society and the concern of the consumers, estrus expression is a beautiful plan B. Because if estrus expression has a genetic component, it does. It has a genetic component. Then we can select so cows show more estrus. And if the cow shows more estrus, it's easier to detect that cow in estrus. We will increase insemination rate and at the end of the day, pregnancy rate, because those cows that show more estrus are also more fertile cows. So this is another quite interesting trait that we are working on.
No, that's really interesting. They're really a good time to see how this evolves and what comes out of it. Let me switch screens here real quick. Two more questions, and we will get to our next presenter. We know that only about 70% of cows are cycling by 60 days in milk. What will we do with these cows without hormonal intervention?
Well, Paul is our expert here. He could unmute and answer. I think he's actually challenging me what I'm saying about the minimize the use of reproductive hormones, right? And probably if we need, if in the future, maybe we can do a combination of genetic selection for estrus expression and minimize the use of, or we use reproductive hormones only in those cows that we need to. And the other one is, have there been studies with quality of feed or? Well, there are studies, right, showing the importance of feeding on production, and there has been a tremendous increase in milk production, right? 60%, let's say, of that is due to genetic selection, but of course, the rest is explained by better management, better nutrition, better cow comfort, and of course, the genetic of the animal provide the ability. Then you need to provide the opportunity, right? Good management and good feed for that animal to observe this ability.
Great. No, and just for our recording here, I'll read back that question. Have there been studies with quality feed or chemical fertilizers correlating with production decreases or increases? Yeah, if you think of more questions, definitely send them in. Otherwise, we'll transition. I will put a quick plug in. I believe it's March 7th. Dr. Penñagaricano is going to be back with us talking about genomics and correlating that with our discussion on feed efficiency. So please join us again for that session as well.